Milk contains proteins, and these are broken down into peptides by enzymes in the body or during food processing. Some of these peptides are bioactive, which means that they have an effect in the body. Here in FHI, we have screened over 1,000 peptides and have identified 75 with a potential health benefit. This short video is designed to demonstrate our work and to show you what research capabilities we have in our centre. In our intelligent milk mining team, we connect all required capabilities and technologies to a very structured, informed, fast and specific feedback loop between the fundamental discovery and generation of new milk components, the identification of bioactive peptides, and the prediction of bioactivity and the final test of bioactivity in specific bioassays. Once bioactivity has been proven on a lab scale, FHI scales the samples to pre-commercial quantities, confirms bioactivity again through the same feedback loop, and adds sensory elements, food structure design, and sometimes encapsulation to the research approach.
At the University of Limerick, we use different types of enzymes that can cut proteins in different places in order to release shorter fragments that are called peptides. We know that different conditions such as temperature and pH can affect the structure of the peptides that are being released from those proteins. We use those different combinations of parameters in order to release different peptides that are then sent out through the FHI team for bioassay screening. In FHI, we have different bioassay teams that study the bioactivity of those peptides, and they also search within these peptides the most potent structures that are responsible for the bioactivity.
Focusing not only on enzymatic hydrolysates, in Teagasc Moor Park we generate bioactive compounds from the fermentation products of food-grade bacteria. Mass spectrometry allows us to survey very many proteins and their hundreds of fragments found in foods. This state-of-the-art technology is a technique which will not only identify the presence of peptides in a medium, but can also measure how much of that peptide is present in a given sample. Bioinformatics provides the computational tools to analyse all the peptide sequences observed in a sample, allowing us to map the peptides in food preparations that benefit health. These computational peptide maps serve to guide the design of better healthy foods enriched for beneficial peptides. The production of hydrolysates in a tightly controlled environment is very different to the production of them in a larger, more commercial size setting. To develop the satisfactory procedure to produce them in this larger scale is known as upscaling, and this work is carried out in Teagasc Moor Park. In this facility, we also upscale fermentates and other milk components to pre-commercial level. This upscaling involves the use of separation technologies and the enrichment of bioactive molecules by membrane filtration techniques. When milk proteins are broken up into shorter lengths, these shorter peptides that are produced have different structural properties to the original milk proteins, and this can also affect their taste and flavour. One of the main challenges for sensory scientists in the food industry is that bioactive peptides often taste bitter and astringent. In FHI, we have a team who work to profile the sensory properties of milk bioactives using trained sensory panels and state-of-the-art techniques, including an electronic tongue. Understanding the properties better can help us to design targeted approaches to improve taste and texture, and can help us to design the most appropriate food matrices for different types of bioactives. The bioactive peptides generated in FHI are likely to be fragile. Potential interaction with other ingredients within the supplemented food, or loss of activity due to heat treatment processes needed to ensure long shelf life and microbial safety of the functional food products, are but one threat to the stability of these bioactives. Once ingested, the harsh environment of the digestive tract, including low gastric pH, digestive enzymes, etc., could result in further hydrolysis of the peptides into inactive compounds. For all of these reasons, One aspect of the research within FHI has been to develop food-grade protective or encapsulation matrices capable of protecting peptides from gastric digestion and to ensure delivery. All of these teams in the FHI Technology work package form our Intelligent Milk Mining team, which is the core of the FHI operations. This team provides the program with peptides, helps to optimize the production processes, and helps to characterize and understand the peptides to improve our knowledge of their physical and sensorial characteristics. This knowledge will enable our industry partners to produce milk peptides, which will have a variety of health-enhancing benefits with high commercial potential.